to introduce Professor Gospodinova, who will be the next speaker on this satellite symposium, and we are very happy, we are very privileged by that Pharma Genesis is giving us the opportunity to have with us Professor Gospodinova. Professor Mariana Gospodinova is a leading Bulgarian cardiologist with over 25 years of clinical and academic experience. She specialized in heart failure, cardiac amyloidosis, cardiomyopathies, and cardiogenetics. She has a PhD in cardiac involvement in hereditary neuromuscular diseases and is also chair of the working group of myocardial and pericardial diseases of the Bulgarian Society of Cardiology. She has been the author of over 100 scientific publications, and she is a fellow of the European Society of Cardiology. The talk that she is going to give to us today is about novel agents to treat cardiac amyloidosis. What are the clinical data and the future perspectives? Professor Gospodinova, please.
Dear Professor Michalis, dear Professor Eftimiadis, dear colleagues, it's a great honor for me to be here. I am really happy to be in Thessaloniki. I like this city. First of all, I would like to thank Professor Eftimiadis for his kind introduction to be part of this very important meeting, and Genesis Pharma for the support, and also to Professor Michalis for his kind introduction. Yes, my task now is to talk about novel agents for the treatment, specifically for ATTR cardiac amyloidosis, and what are the clinical data and the future perspectives. We are very well aware of the massive and significant progress in the diagnosis and the treatment of this rare in the past disease, and now not so rare. This is like exclusion from all other rare diseases where we can see such a significant advantage. These are my disclosures. You are very well acquainted with this, but just as introduction, we know that there are two main types of cardiac amyloidosis: light chain amyloidosis, that is predominantly oncohematological disease, but very frequently the cardiologists are first to meet these patients and to put the diagnosis, but the management of these patients is conducted by the hematologist. And the other most common type of cardiac amyloidosis is transthyretin cardiac amyloidosis, and you know that there are two types: hereditary type that is caused by pathogenic variant in the transthyretin gene, and this is a multisystemic disease affecting not only the heart but the peripheral nervous system, and sometimes ocular involvement, leptomeningeal involvement, or kidney involvement. The cardiologists are much more interested nowadays, I think, in the wild-type ATTR cardiomyopathy. It affects many people, predominantly male patients above 65, and the heart is the target organ. Of course, there are some other organs affected, mainly soft tissues. Here on this slide, you can see the process of amyloidogenesis, the therapeutic targets, the available and potential future therapies. Transthyretin, the pathogenic protein, is produced by the liver, circulates as a stable tetramer, but because of some changes in the genetic changes or aging, this stable tetramer dissolves into monomers, which are amyloidogenic. They form fibrils, and they are deposited in different organs and tissues, and they change their structure and function. And the targets of the therapies, they follow the steps in this amyloidogenesis. If we start upstream from the production of the transthyretin in the liver, you see that the gene silencing is the main concept for the reduction of the production of the amyloidogenic protein. And we know two types of drugs that target and cause the gene silencing. First of all, I'll mention the small interfering RNA, because Here is the drug the only drug from this class that is approved for the treatment of ATTR cardiomyopathy. This is a second generation small interfering RNA, vutrisiran. We know that we have a great trial showing the significant better outcome in patients treated versus placebo. And this is second generation, as I said. It is administered subcutaneously in every 3 months. The first generation drug, patisiran, is approved for genetic disease, for ATTR polyneuropathy, and it is administered intravenously, which is some of a problem for many patients. Here I will mention that at the moment there is a clinical trial conducted with third generation small interfering RNA. This is nucresiran, but it will be administered subcutaneously 2 times per year, every 6 months, but it's in the process of investigation. The next, here we must mention the gene disruption. We know that there is a phase 3 trial with NextZ. This is using CRISPR-Cas9 technology. We were very enthusiastic, but we know that it stopped not permanently, temporarily, because of a side effect, one death due to liver problems. The TTR stabilization, we know that this was the first target and the first drug approved for the TTR stabilizer. This was small molecule tafamidis in 2018 after the ATTRACT trial, and it was after that approved by the regulatory organs. Until recently, this was the only drug for the treatment of ATTR cardiomyopathy. But now we have vutrisiran, and the other drug that is also approved for ATTR cardiomyopathy is vutrisiran, another small molecule, another stabilizer. The trial was reported and published last year. Now the drug is approved by the international regulatory organs, and maybe it has better stabilization than tafamidis. This is the promotional finding. Our expectations are the last to the right of the figure. You can see that now there are trials conducted on monoclonal antibodies that potentially could clear the existing amyloid and improve the structure and the function of the heart. There is a review paper published in European Heart Journal this year. It was very interesting, and it promotes the hypothesis that amyloidosis is not a new hypothesis, but in the light of the new therapies coming, amyloidosis is the product of imbalance between production and clearance, and amyloid deposits exist in a state of dynamic equilibrium dependent upon the rates of ongoing amyloid formation and natural amyloid clearance. In the past, we thought if we have amyloid, it's there, we can't do anything about it. But now, using some imaging data and other histological data, we see that it's maybe a reversible process, but the reversible process is much slower than the deposition of the amyloid. That's why we need drugs that can help to reverse and to clear these amyloid deposits. And here you can see 4 theoretical models. If we know that the process of deposition of amyloid is much faster than the process of clearance, and if we don't intervene, if we don't give drugs, there is a progressive increase in the amyloid infiltration. But if we give drugs that decrease the production or stop the production of amyloid fibrils, then we can slow the deposition of amyloid. Another scenario, if the patient has faster clearance, the natural ability to clear the amyloid is higher, then we can see even downslope of this deposition of amyloid. And the patients who are slow metabolizers, it's not the correct term, but then if we give monoclonal antibody, then also we may convert the slope down, so to decrease the deposition of amyloid and also to extract the amyloid from the tissues. It's very, for people who are not in the field, they are very curious how we promote drugs that stabilize the transthyretin. So we increase the transthyretin, and at the same time, drugs that silence the transthyretin or lower the production of the transthyretin. We see that both hypotheses work, having in mind the clinical trials, and there is no comparison which work better, and there are no also some issues about the side effects of both drugs. About the reduction of the production of transthyretin, this hypothesis is also supported by some findings in other types of amyloidosis, for example, AA amyloidosis, AL amyloidosis, and we see from other trials and other real data that when we decrease the amyloid precursor protein, we have better prognosis. This is also seen in ATTR polyneuropathy in HELIOS-A study. We see that when we knock down the production of transthyretin, then we have increased MNIS score, which is score that shows improved polyneuropathy. Here you can see the mechanism of action of RNA interference that is the basis for the production of these drugs. This RNA interference, it mimics the natural RNA interference mechanism to decrease production of the target protein. This is a process that helps to regulate gene expression. And if we have synthetic small interfering RNA, it is double-stranded, it crosses the cell membrane, and in the cell, it enters the RISC. This is ribonucleic acid-induced silencing complex, where the passenger strand is removed, and the guide strand, it is paired with the messenger RNA, and it disrupts the messenger RNA, and in this way, it decreases or stops the production of the pathogenic protein, in this case, the transthyretin. And nowadays, we also ask the question, which drug for which patient, which drug is better? Could we combine drugs? We have no answers for these questions because we have no head-to-head comparison trials. And we see in every trial that there is significantly better results in patients on treatment compared to placebo in all three trials. Here are the primary endpoints in ATTRACT with tafamidis, all-cause mortality and rates of CV-related hospitalizations. In attribute cardiomyopathy, we have a bunch of final endpoints: all-cause mortality, cumulative frequency of CV-related hospitalizations, changes from baseline in NT-proBNP and 6-minute walk test, and HELIOS-B composite endpoint of all-cause mortality and recurrent cardiovascular events. But if we compare the baseline characteristics of the patients included in the different trials, we see that they are not equal. So we can't compare the results of these trials because we can see that the contemporary population is healthier because of early diagnosis and the concomitant treatment with other heart failure drugs, for example, SGLT2 inhibitors. We can see that the patients in class 2 and 3, in class 3 decrease, and in class 1 and 2, they increase in the trials that are more recent. And also, the quality of life is better in the contemporary population, and the NT-proBNP is lower. It's the highest in ATTRACT trial. So these trials are could not be compared. And now my task is to present the results of HELIOS-B trial that was used for the approval of vutrisiran for ATTR cardiomyopathy. It is a phase 3 randomized double-blind outcome study to evaluate vutrisiran in patients with ATTR cardiomyopathy. 655 patients were included with wild and variant type. Of course, wild type much more common than a variant. Confirmed cardiomyopathy and heart failure, and some requirements for 6-minute walk test and NT-proBNP. And it's interesting that approximately 40% were on baseline tafamidis treatment. Exclusion criteria for NIHA class 4 heart failure, and also NIHA class 3 and NAC stage 3. Also some requirements about polyneuropathy and other disease-modifying drugs. Which were the primary endpoints? I said that was all-cause mortality and recurrent cardiovascular events, but you can see that they were explored in two populations. One population was on monotherapy, only on vutrisiran, and the other was on vutrisiran on top of tafamidis, because it was approved for treatment. It was not possible to have placebo. The secondary endpoint change from baseline to month 30 in 6-minute walk distance in KCCQ at 42 months of mortality, and also NIHA class. Well, I said that, but we see that this includes contemporary patient population, and we have substantial use of tafamidis baseline, 40% in both treatment arms, and also we have drop-in among the monotherapy population in 21 and 22%, above this 40% in placebo and vutrisiran, and also a significant drop-in of SGLT2 inhibitors. Also, we can see that with vutrisiran we have a very early and stable knockdown of the transthyretin, about 87%. We know, if I said this here, the hypothesis is the lower the better. And the HELIOS-B met all primary and secondary endpoints in both in the overall population and in monotherapy population. We see that the primary endpoint, the hazard ratio in the overall population was 0.72, and in monotherapy 0.67. And also the all-cause mortality at month 42 has a ratio in overall 0.65 and in monotherapy 0.66. So we have significant better results in the treated population in both arms compared to placebo. And here is the graphical illustration of these results. We have statistically significant reduction in the composite all-cause mortality, and we see that the curves diverge after approximately 6 months. And these results were consistent across all pre-specified groups according to age, 75, according to tafamidis baseline, about the type of ATTR, whether variant or wild type, NIHA class 3 or less, and the baseline NT-proBNP levels in both evaluated groups of patients. Also, here we can see that the death, all-cause mortality or death from any cause through 42 months was significantly in favor of the treated population, also consistent in all pre-specified groups. And also the secondary endpoints were also with clinically significant difference with placebo, having 6-minute walking distance, and the quality of life questionnaire, and also the NIHA class. Also, there was some exploratory endpoints, for example, cardiac biomarkers. We know that they have relation with mortality, and we see 32 relative reduction of both NT-proBNP and troponin I at month 30. And also, we have positive trends in the cardiac structure and function. I won't specify which parameters were explored. Also, reduce cardiovascular mortality at month 42 with 33% in the overall population, 36% in the monotherapy population. The cardiovascular events. And most importantly, there were no the safety summary was positive for the treatment group. So the incidence of adverse events in the vutrisiran group was similar or lower than the placebo group. And they were consistent with previous trials, for example, HELIOS-A, which is for hereditary ATTR polyneuropathy. As in other trials, after the conclusion of the double-blind period, an open label extension study was planned and conducted in order to have longer evaluation of the patients, not only for the positive results, but for the safety reasons. And we can see that this long-term vutrisiran treatment significantly reduced the risk, the composite endpoint of all-cause mortality and cardiovascular events, with 37% in the overall population and 42% in the monotherapy population. If we take only the all-cause mortality, we have 37% reduction in the overall population and 39% in the monotherapy population. Also, here we also, in this extension period, we have no safety concerns. The difference between the two groups was in favor of the treated group, not of the placebo group. And here I'll show you one of the abstracts from the latest American Heart Association scientific session 2025 by Mariana Fontana. We know that she is expert in amyloidosis and also in cardiac magnetic resonance evaluation of cardiac amyloidosis. And here are the results of a small group of patients that were part of the HELIOS-B trial, but they are following also their algorithm of following the patients. And here, by magnetic resonance imaging, we see that the treatment with vutrisiran was associated with statistically significant and directionally favorable changes in multiple measures of cardiac structure and function compared to placebo. And you can see to the right the diverging curves in left ventricular stroke volume and left ventricular mass in patients treated and not treated. And we know that the extracellular volume is one of the imaging parameters we can use for following the progression or regression of the disease and the effect of the treatment. And we can see that in 22% of the patients, it's a small group of patients, I must say, they find amyloid regression in patients treated with vutrisiran against placebo. You can see baseline ACV 61%, after 36 months 55%. These are two examples, 54, 48. But there was amyloid progression observed in 63% of the placebo patients, for example, from 49 to 55%. This is about the extracellular volume, and this finding maybe supports the hypothesis that effective suppression of TTR production can shift the balance between amyloidogenesis and clearance, permitting amyloid regression and cardiac remodeling. We know also there are publications about the first generation small interfering RNA patisiran, also with the extracellular volume. And there are other case reports that are published about the amyloid regression, and this supports the hypothesis of this dynamic equilibrium that is either due to the natural immune system of the patient, or it can be also helped with the drugs that influence the TTR production. And here's one slide for Bulgarian experience with vutrisiran in patients with hereditary ATTR amyloidosis with polyneuropathy and cardiomyopathy. In our country, the drug is approved only for ATTR polyneuropathy at the moment. Now we applied for in the National Health Insurance for approval for cardiomyopathy, but still the cardiologist can't prescribe vutrisiran, only the neurologist. And here you can see that we have at the moment from October 24, when it was approved for polyneuropathy, till now, 57 patients. 16 were switched because of progression on tafamidis. 25, they were changed from patisiran. They were on third generation drug. 7 were switched from inutersen because of adverse events. We have a lot of adverse events with inutersen. Also 4 de novo patients, 6 from HELIOS-A, because we participated in HELIOS-A. This is for ATTR polyneuropathy, and 1 patient was deceased. And in conclusion, we can say that RNA interference therapeutics offer multiple benefits: rapid knockdown, consistent knockdown, target detection with low potential of toxicity, high metabolic stability, long duration of action every 3 months and potentially every 6 months, and specific gene silencing and down-regulation of the target protein. And with this, I finish my talk. Thank you for your attention.
Thank you very much, Professor Gospodinova, for this fascinating talk and about new agents and how they transform the treatment of the heart, and that show that heart and cardiologists were not just plumbers and electricians, but were doctors, still doctors as well. Do we have any questions? Yeah, Professor Vlachopoulos and then Professor Naka.
Mariana, excellent presentation as expected. I have a question. The results of HELIOS-B were very impressive, and if you dissect the results for the monotherapy, it means that the drug, because they had a great percentage of concomitant treatment, if you dissect the results, the monotherapy showed equally good results. So I would reverse the point of view and say there's no need in combining the 2 treatments, because for now it is very difficult due to the expenses, but in the future it might be the combination of 2 different acting drugs could be an idea for, especially for people that do not respond so well.
Yes, if we think about, as doctors, if we see patient that progresses, we will want to help him, and if he progresses on 1 drug, we will think about either switching or adding, maybe after adding. But at the moment, we have no data from, we have no such trials to compare. Now this is, yes, we have a large number of patients on both therapies, but no statistically significant difference in the monotherapy population and in the population on the families. So I'm not sure I know the answer whether we could help our patients with adding the second drug, but our intuition that we must do something and we will act. But because we know the prices, I think that it won't be in the national insurance officially.
Professor Naka.
Thank you for a very informative and educational talk. I have 2 questions. Looking at the rate of stopping the production of the protein, I would probably expect a greater response, a greater clinical response. Do you think that addressing better functional class patients, not so much advanced disease, even NYHA1, for example, that would be more effective in treating patients?
Treating earlier, you mean?
Earlier, yeah. If you find them, obviously, if you diagnose them
yes,
earlier.
Yes, of course. I'm sure that if we treat patients earlier, we will have better outcome. Yes, the problem is to diagnose patients earlier, but if after that we must say, if we find, for example, positive scintigraphy in a patient that has no heart
Symptoms.
failure, no symptoms. Shall we start treatment in order to prevent? Because we know that many, for example, elderly patients, they have amyloid deposition in the heart that is there. It increases with age, but they die from something else. They don't have heart failure. So yes, I think, for example, in the hereditary, what we do, if we find not symptoms, if we have signs of the disease, either with very sensitive neurological tests or with cardiac tests, for example, bone scintigraphy or magnetic resonance, and the patient feels healthy, we start treatment.
Well, we discussed, that's interesting because we discussed this in the morning session from the Greek cohort in Crete. And in Greece, in the cascade screening, we would not start treatment.
Okay,
that's what
we start.
You start. Yes.
If we have signs from polyneuropathy or cardiomyopathy without symptoms, because we see that when we start treatment in symptomatic patients, most of them progress. And we know that there are trials, it's with another drug that is conducted at the moment in carriers. It's also about the hereditary type in healthy carriers. And the result will be interesting and will confirm or reject the hypothesis of treating, in fact, healthy patients. For the hereditary, this is my opinion. About the wild type, yes, I think that if we have definite diagnosis and we see organ involvement, it's better to start treatment. This is my personal opinion.
And another question on your experience from the Bulgarian cohort. You had 16 patients who were on tafamidis, and you saw some evidence of progression.
Yes.
Can you share with us what was this progression based on, and whether you switched them from one agent to another, or you gave both treatments together?
So every 6 months we follow the patients with hereditary amyloidosis from neurological and cardiac point of view, and we perform specific tests. The neurologists perform their tests data from pseudoscan and polyneuropathy and score. I'm not sure, and also with about the cardiac involvement with cardiac biomarkers, ECG and echocardiography, and we discuss every patient, and depends on what is the cardiological observation and data and the neurological. We decide that the patient progresses and we shall switch to another therapy, but we can't give both drugs together because it's not possible. It's not in our national insurance system. It's not permitted.
Any other questions? So let's summarize a bit. This looks like a first-line drug treatment.
Yes.
It's not just to wait and see and then add it.
There is no reason not to be to first-line therapy because the results are equal or better to other therapies. And these are our, it's in our country, in the hereditary type, is first-line therapy also, and also we applied for first-line therapy in the wild type.
We don't have data that adding the two treatments, although the theoretical models support it, we don't have clinical data to support this way of management of patients to add two treatments, one of stabilizer and one of stopping the production. That's what I understand. What impressed me a bit is that we have signs of remodeling, that it's not a reversal of the progress of the disease. It's not just stabilizing it, and that's something which is intriguing. If we have the real data that will show that the disease regresses, we have a regression of a disease, then that will be a very good thing for our patients, and that's a very sort of nice finding.
I think that there is real regression, but it's not for everyone. As Professor McKenna said, every patient is different, and when, there are many factors, genetic, epigenetic, modifier, in every patient immune system, and not every patient, in every patient the result is the same.
Okay, so
and also it depends on at which stage of the disease the treatment has been started.
Okay, it's a new road, just started, new fancy treatments, and I'm sure that we'll see very nice, very different things in the future that will change the whole atmosphere and the whole management of our patients. Thank you very much. Thanks a lot.
As Professor Rapezzi, every time I remember him said, it's a fascinating disease.